And thank you, Professor Yoon, for that wonderful and really informative talk that you just gave. And I'm sure like this would have motivated many of the young researchers and students who have joined this talk and probably guide them towards starting their own research career in similar topics. And we have a list of questions that we have curated, and I'll start off with the first one. So this is from one of the audience member. It's a more targeted question, maybe a bit more scientific. So can you please suggest an approach for the stability of carrier systems such as drug-loaded nanoparticles used in vivo applications? And he or she has already cited an example, like in literature, we encounter DLS measurement in medium containing serum, but serum protein prevent correct interpretation. Do you have any advice at this point?
Yeah, well, this can be the more technical, actually, question for me, honestly. So maybe, yeah, this can be the problem, but maybe I cannot answer very clearly for this one, since, yeah, yeah, certainly serum protein can cause a problem, I think, to you mean measure the DLS measurement.
All right, Professor Yoon, I'll move to the next question then.
Okay.
What is the minimum requirement of lipophilicity of probe for photodynamic therapy?
Well, if you do not use you mean carrier, then lipophilicity well, this can be similar to the just drug design, I think. So lipophilicity of the photosensitizer is probably similar. So you have to think about the clearance from the body. And if you do not use that other lipid nanoparticle or polymers, then so lipophilicity should be similar to that of the, you mean, drug design or even process prof, so which can be uptake by the cell. So I think that's my answer, but maybe not really good answer for this one.
I'm sure like this will be helpful for the person who was doing that research and who posed that question. But I'll move to the next one. So which, according to you, the phototherapy modality that is more effective? Is it PDT or PTT or a combination of both?
Well, both of them has its own advantage. Of course, PTT has relatively even better penetration depths than PDT, but so I think it really depends on. So, but usually PTT requires higher, you mean, laser energy compared to the PDT. So I think it depends on. But if you can have you mean both synergistic effect, maybe better because you can do the fluorescence imaging, but the photoacoustic imaging is also penetration depth is better than the fluorescence imaging, so you can probably see both effect from PDT and PTT in this case. Also, if you can control the selectivity for the PDT and PTT, that would be also helpful and interesting too, I think.
Thank you, Professor Yoon, for that answer. I'll move to the next question. So an audience member has posed a question. Can you please shed some light on the application of carbon rods as fluorescent probes?
Well, I don't have much experience with carbon dots, but if the carbon does also does not have a serious problem for the safety problem, then yeah, it certainly has advantage in potential for the imaging probe and as well as photosensitizers. So because yeah, it can be made nowadays various amino acid or various species. So yeah, carbon certainly has a potential. Yeah.
Thank you, Professor, for the answer. I'll take another one. So it's more related to the work that you just presented. So can you please tell us why MANIS, that's the probe, I'm guessing, disassembles in the cancer cells, but stays assembled in normal cells, and hence, like, there is no fluorescence in the normal cells.
Oh, many S?
Yes, yes, Professor.
Let me go back. Sorry. It is not actually, it is from the, as you can see here, it is from the calcine AM and also the ROS probe. So the answers for that is this process comes from the ROS probe, so which supports that this probe is generating the ROS under hypoxic conditions. So I hope that's the answer for the question.
Thank you, Professor Yoon.
Mm.
I'll take another one. It is also related to the work. If we administer the albumin trap phthalocyanine derivative intravenously, will the interactions with serum albumin interfere with its accumulation in the tumor tissues?
Yeah, albumin is known as maybe passive and also both you mean the glycoprotein 60, I think, yeah, is the one of the active and passive targeting is possible for the albumin. So that's the reason why albumin is actually known to target the tumor. So we just use that advantage of albumin by trapping by the albumin and to treat the tumor.
Thank you, Professor, for the answer. So you also talked about one of the rhodamine-based probe that was commercialized by your group. So could you also shed some light on the timeline that is followed from lab to clinical trials to finally being commercialized?
Well, for the compound, so it was just commercialized as just a process probe for HOCl. So people can use it for research purpose, or people can use it to detect the diseases using this probe. But this compound, specific compound, was not FDA approved or something like that. So we just commercialized this one for the HOCl probe. And in my opinion, for the process probes, since we are using relatively small concentration, small amount compared to the regular drug, so that's maybe one of the reasons we already have few, process probe FDA approved ones too. So but this one was just commercialized for that purpose. That's all. Yep.
All right, Professor Young. I'll move to the next question. So could you please elaborate on the reason for selectivity of NBSN towards gram-positive bacteria?
Yeah, for this one, the core compound was published by the Xiaojun Pang's lab in Dalian in JAX. So so the selectivity of the generation of ROS, for example, NBS and MBSE, is previously reported. So as I mentioned, that the selectivity comes from the different membrane of this bacteria. So as I mentioned, this singular oxygen is toxic enough to kill both gram-positive and gram-negative bacteria. On the other hand, this superoxide radical anion has a moderate reactivity compared to singular oxygen. And this can be, of course, can be converted to highly toxic hydroxyl radical only inside of the bacteria. So this membrane difference is also combined with the ROS generation difference between these photosensitizers are coupled in this case to cause or to induce the selectivity for the gram-positive bacteria. So that's maybe the one of the approach which can design the gram-positive bacteria. But then again, whether this selectivity for the gram-positive, gram-negative is really important or very effective bacteria, a killer photosensitizer is effective. That's something we still have to think about. But anyhow, the selectivity comes from this difference.
All right, Professor Yoon, thank you so much for the detailed answer. So the next question, so this is about, it's a general question about photosensitizers and how is the scenario right now in terms of the clinical application and how your work can actually be applied for practical applications or clinical applications for that matter.
Yeah, in our lab, we are approaching two different ways, as probably some other labs are trying. So one is using FDA-approved photosensitizer and try to improve the selectivity or try to improve the enhance the generation of ROS or so. On the other hand, the other approach is, of course, that design and synthesize new photosensitizers, especially for the type 1 process. So for that one, as you know, it is a long way to go to solve the safety problem. But if the property is really, good enough, then and also we can probably can continue to the clinical trial. So right now we are using some BSA-related photosensitizers, protein photosensitizers, and also the compound synthesized from our group already, investigated by the company. So hopefully we can see some progress within a few years for this purpose.
Thank you, Professor Huyon. We are almost on time here, and if you are comfortable, can I throw a few more questions at you, because we got quite a few from the audience members, but only if
Yeah,
you're.
that's fine.
Thank you. Thanks a lot. So it's also a very specific question. Is the vinyl dicyano compound safe for bioimaging? Will there be any chance of it to metabolize with time and become toxic?
Which one was it? I'm sorry.
Vinyl dicyano.
Oh, viral dicyano.
Right.
Oh, well, yeah, I'm not 100% sure for that. So
Okay.
yeah.
So is there any benefit of using green photosensitizers like anthocyanin or beta carotene dyes, in your opinion?
Green photosensitizer. Green?
Mostly, I guess what it means is bio-based, or maybe coumarin derivative as well, or some plant-derived photosensitizers.
Well, for the photosensitizers, like fluorescence probes, NIR, NIR or NIR2, maybe better. You mean if you think of the penetration depths and also the damage of the light. So in that sense, if you can make a longer wavelengths absorption photosensitizers, definitely has a better chance. On the other hand, two-photon probe or two-photon photosensitizers for PDT. So, well, if I'm not quite sure exactly, you mean which just core is the best for the purpose. But the fluorescent-based one already FDA approved. So few groups are, you mean, using this FDA approved core to with activity for the enzyme or other ROS derivative, and which already been FDA approved and is used. So coumarin also can be one of the good candidate if you can make it, you mean not blue one, a little bit longer wavelengths probe or photosensitization.
Thank you, Professor Yoon. Kunal, do you think we can squeeze in one more general question? You're on mute, Kunal.
So maybe we can sign off if Professor Yoon has some remarks for our audience members, because we have a lot of students who come in and there have been some generic questions, so we can have this last take on that.
So well, thank you so much for your kind attention and attendance for my talk. Maybe I'm just one of the researcher in this field. So I've been working for this for us probes and photosensitizer for the last 20 years or so. So hopefully some of the approaches we published can be helpful for the your research, and hopefully you can design and make really useful and very interesting photosensitizer with fluorescence probes in the future. And I hope big success in your future career. Thank you so much.
Thank you, Professor Yoon. So since we have, I think we there's like such a load of questions that have come in. We have not been able to cover many of them. So would it be okay if we send some of the questions your way, and whenever you have time, you can reply to us so that we can
Sure.
share it with our attendees and registrants? Thank you.
Sure, definitely. Thank you.
Perfect. Okay, so with that, we'll close the session. So thank you everyone for joining us today. This brings us to the end of today's session. We hope that you have enjoyed this broadcast. We invite you to view the edited recordings for the past events in the ACS Science Talks library. Our colleagues have already posted a brief survey for you to take. I hope that you've seen in the chat. Please take that survey. It helps us to get your feedback, and so that we can continue to improve and serve you better. On behalf of all of us at ACS, thank you for joining us today. We hope to see you again soon for more ACS events. Until then, stay safe and healthy.